We need to look now at the concepts of diffusion and osmosis. This is how fluid moves in and out of the cell. So it's called membrane transport. So we're following what is called the fluid mosaic model of membrane structure. So that the cell membrane is a barrier, but of course it's not a barrier to everything because nutrients have to get in and waste products have to get out. So the permeability, or how things move in and out, is depending upon the structure of the membrane and what's present in the structure. So a membrane can be impermeable, which means nothing gets in and out. It can also be freely permeable, which means it allows anything to move in and out. Or it can select what it wants to move in and out and be selectively permeable. So it can restrict the movement of certain items in and out of the membrane. So membrane transport can require energy. If we're looking at selective permeability, so it allows some materials to move freely but restricts others. And the restriction can be based on the size of these materials or the electric charge. So remember, the electrolytes are charged particles. So it may depend on whether it's a charged particle being positively or negatively charged, the actual molecule shape, and also whether it is lipid soluble. Remember, in the first lecture we spoke about the membrane, the plasma membrane, being a phospholipid membrane. So it is soluble to lipids, soluble to fats. So it will allow fats to move through. But if it will allow fat to move through, it will not allow water soluble to move through. So membranes can be, transport through the membrane can be active. So if it's active, we'll look at that A in active, and it requires energy. It requires ATP. If it's passive, then it doesn't require energy. Passive movement through the cell membrane is known as diffusion. You can have carrier-mediated transport, which can be passive or active, and then you can have vesicle transport. Remember, we looked at the lysosomes. They produce vesicles, and that the solutions within the vesicles are then pushed through the membrane, and that requires energy as well. So let's look more closely at diffusion. So all molecules are constantly in motion. And molecules in solution move randomly. They bounce around against each other. And that random motion causes mixing. And the concentration is the amount of solute, so the amount that I put into the solvent, which is the solution. And the result is what is called a concentration gradient. So if we have more solute in one part of the solvent to another. That all sounds very confusing. Let's look at a diagram. So here's our diagram. So we've got the same beaker after a time delay. So here we are adding a little cube of colour, which is going to dissolve in the solution. So the little cube of colour is called the solute, and the water, or what is going to be dissolved in, is called the solvent. So you can see that the solute, once it is added to the solvent, the molecules of the solute begin to be released and bounce randomly against each other until there is a concentration which is evenly distributed through the solvent. The example which comes to mind, of course, is if you've got a cup of tea and you add grains of sugar to your cup of tea. You can stir it around, and that will increase the distribution, or how rapidly it will dissolve, until the sugar solution has moved right through the tea, and when you take a sip you get sweet tea. So I'm asking you here to pause the lecture for a few minutes to view the media clip called Diffusion and Cell Membrane Barrier, which is found in the Week 5 study plan. So this will help reinforce the concepts that we're talking about, and I'll see you in a few moments. So here we are back again after you've looked at those two short media clips about diffusion and the cell membrane in fluid movement. So here we are looking at diffusion and osmosis. We've got to look at the factors that affect diffusion. So the distance the particle has to move, the molecule size, the smaller the molecule, the faster it is, and also heat. So if we look at an example, so if you've got a large lump of sugar and you put it into a saucepan of water, it's going to take a long time for that large lump of sugar to dissolve. If, on the other hand, you got some icing sugar or caster sugar and put it into the saucepan of water, then that would, because it is a smaller size, it would happen faster. If you then took the saucepan with the water and the sugar and added and put it on the oven top and added heat, that would increase the rate at which the sugar was dissolved into the water, so it would speed it all up. The term that you need to come to grips with is concentration gradient. So concentration gradient is the difference between high and low concentration gradients. And the concentration gradient refers to the amount of solute. So a high concentration gradient has a high amount of solute in the solvent compared to a low concentration gradient which doesn't have very much solute in the solvent. Also need to know that electrical forces, when we think about that positives, so opposites attract. So a positive and a negative charge will be attracted to each other, but two positive charges or two negative charges together will repel each other. So you can prove that by getting two magnets. Remember, a magnet will have a positive end and a negative end, and you can try and push the two positive ends together, but they will repel each other. But if you turn the magnet around the other way and had the positive and the negative end together, then they will attract each other and the magnet will stick to each other. If we look now at plasma membranes and diffusion, it can be simple or channeled. If we look at materials that diffuse through the membrane by simple diffusion, we're looking at lipid-soluble compounds. So remember, the cell membrane is a biphospholipid membrane, so it has fat within it and it allows fat-soluble compounds to move through it. Also, what can move through it are dissolved gases like oxygen and carbon dioxide. If we look at cell-mediated diffusion, such as water needs to move into the cells, it has to move through by channel-mediated diffusion. So this is dependent. So channel-mediated diffusion is dependent upon the size and the charge and the interaction between the channel. So it should be able to leak through the channels. So here's a diagram of what I was talking about. So here, if I just describe the diagram, we've got the purpley blue area, which is the extracellular fluid. So that's outside of the cell membrane. At the bottom part of the diagram, we've got the cytoplasm, which is that sort of buffy orangey color. That's inside the cell. So within it, separating it, we've got that biophospholipid layer, which is the cell membrane. Remember that the round bits on the outside are the hydrophilic ends, the water-loving ends, and the tails that are facing towards each other, those lines that are lined up against each other, are the hydrophobic ends. Now, we've got fat molecules, which are lipid soluble, and they can move freely, passively, through the cell membrane because they're fat soluble. But we have other parts of the cell membrane known as channel proteins, and they allow water-soluble molecules to move through. But you can see there that it's not just the water that's moving through, the green bit being the water. You've got some other things moving through as well, and they are charged particles or electrolytes. So we'll look more closely at those in a minute. Osmosis is a special sort of diffusion. So osmosis is only the diffusion of water across the cell membrane. Osmosis is a really important process that you as a registered nurse need to understand, because you'll have an understanding of this when your patient's blood results come back as to whether they are overhydrated or dehydrated, or whether their electrolytes are low or high. So osmosis is the diffusion of water across the cell membrane. More solute molecules lower the concentration of the water molecules. So if you have high solute molecules, that means you have lower water molecules. But the membrane must be freely permeable to water. So the cell membrane must be freely permeable to water and selectively permeable to the solutes, which means the solutes can't move through freely. The water molecules diffuse across the membrane towards the solution with the more solutes, trying to balance it out. And that means that the volume of water will increase from one side to the other. The water will move from where there is a low concentration of the solute to the higher concentration of the solute, as the water will try to balance the concentration of the solute. So here we've got a diagrammatic representation of it, and in our laboratory we'll also have an example for you to observe over the 2 hours of the labs. So if we talk about it, if we look at number 1, you can see that there is a horseshoe of sort of a loop of fluid, 2 sides to it, A and B. If you look at A, we have the small water molecules. The join between A and B is what is called a selectively permeable membrane, which is permeable only to water, not to the solutes. So we've poured our solution A into the A side and the same sort of solution, but less of the water, so a more concentrated solution into side B. Over time, what you can see is that what happens is that the volume of water on side A will decrease as water by osmosis will move through the selectively permeable membrane to try and balance the amount of water on both sides, so that there is an equal amount of water mixed with the amount of solute. If we look at number 3, what can happen is if we apply a force or a weight in this diagram, we can actually force or control the amount of water that can move through. This is important when we look at the extracellular fluid, the intracellular fluid, and the intravascular fluid. So the fluid within the blood vessels, the fluid outside of the blood vessels but not in the cells, and the fluid inside the cells. So here it is again. 2 solutions containing different solute concentrations are separated by a selectively permeable membrane. The water molecules, the small blue dots, begin to cross the membrane towards the solution. The solution with the higher concentration, which the concentration is the pink dots. So if you counted the pink dots on the A side, you would see that there would be more on the B side. And the water will move from the A side to the B side across the selectively permeable membrane. Here, what happens is that we have equilibrium. So the solute concentration on the 2 sides of the membrane are equal. So if you counted the volume of water has moved to create an equilibrium, and you can see that there's a dotted line in the middle, which is the original level. So it's only the water that has moved, not the solutes, because they're too big to move through. You can see the diagram shows that the little gaps in the membrane are only big enough for the little blue dots representing the water to move through, but not the big pink dots. And here, osmosis can be prevented by resisting the change in the volume, and this is called osmotic pressure of B, and is equal to the amount of hydrostatic pressure. And that means that it stops the osmotic pressure flowing. So it stops water moving, even though it wants to move through. So osmosis, special case of diffusion. Osmotic pressure is the force of a concentration gradient of water, and it equals the force, and it's equal to the hydrostatic pressure needed to block the osmosis. So this is important when we look at red blood cells and fluid movement. So osmolarity and tonicity. So the osmotic effect of solution on a cell. So if two fluids have equal osmolarity but different tonicity, then there can be movement. If the solution is isotonic, iso meaning same, tonus meaning tension, then the osmotic flow of water in and out doesn't occur because the solution is the same inside the cell as outside of the cell. So this is important for you as registered nurses because you will be infusing intravenous fluids into your patient, and you need to know whether these fluids are isotonic, so the same amount as the same sort of solution as the solutions within the blood, like the same amount of salt, or is it hypotonic, and that means that it is less than the body's normal level. So this is hypo meaning below, has less solute than, and loses water through osmosis. So water moves away through osmosis. If it is hypertonic, which means it is above the tension, then water moves in and it gains water by osmosis. So let me show you a diagram of red blood cells, and it will make more sense in a moment. So a cell in a hypotonic solution will gain water. That means that water will move into the cell, and in some cases it will actually rupture the cell membrane. In case of a red blood cell, we will get hemolysis, where the red blood cells will hemolyze and rupture, and the contents, or the hemoglobin, will be liberated out into the environment. If, on the other hand, we look at a hypertonic solution, where there is more solute outside of the cell, then water will move outside of the cell to try and balance the concentration gradient. This will result in crenation of the red blood cells, and they will get a sort of crinkled, shriveled appearance. So if we look at the next slide, that'll become a little clearer. So on the far left-hand side, we have a solution where it is isotonic. And you can see that the red blood cell remains the normal size, and fluids can move in and out without any problems. Water can move in and out, but there's no concentration gradient. On the other hand, if we have a hypotonic solution, where the solute on the outside of the solution is less than inside the cell, then water will move inside the cell. And that's where the red blood cell that did look like a donut without a hole in it is now looking like a red round ball. And this can be caused to excess, where that red brown ball will burst and rupture, called hemolysis. And the contents of the red blood cell will be liberated out into the environment. On the other hand, if you then have the example where you have a hypertonic solution, where the outside of the cell is higher in solute than the inside of the cell, then water will move outside of the cell. And you can see here that same red blood cell in a hypertonic solution will become crenated. If we looked under the microscope, which is what you'll have a chance to do, you will see normal red blood cells, and then you will also see crenated red blood cells. So just going through it again. So in an isotonic saline solution of 0.9% of normal saline, which is the same solution of salt as the body, there will be no movement. The red blood cells will quite happily sit in this solution and not be damaged. If, on the other hand, we put it into a saline solution which has less salt than inside the red blood cell, then water will flow into the red blood cell, and the cell membrane will rupture or lyse, called hemolysis. The final one is in a hypertonic, so a high salt solution, and here water will move out of the red blood cells and the red blood cells will shrivel up and become crenated.